What a waste!
Let us first look at how proteinaceous wastewater has become an increasing environmental problem. Wastewaters discharged from food processing industries contain a high organic load of proteins. The discharge of proteinaceous wastewater into water bodies without remediation can cause severe water pollution and eutrophication. In our project, we identified the possibility of utilizing the astringent properties of mangosteen to precipitate proteins. Astringency refers to the bitter and dry sensation in the mouth and tongue, caused by tannins which bind to and complex with salivary proteins to form precipitate. High concentrations of tannins can be found in many fruit and tea wastes. This has great potential to be applied in proteinaceous wastewater remediation as an eco-efficient method of using waste to treat waste. In this study, two properties of astringent fruits and teas were assessed. A. Their ability to precipitate proteins from protein solutions for removal, and B. To reduce the turbidity of milk solution in simulation of turbid wastewater. We hypothesized that the rind of mangosteen is most applicable for use in proteinaceous wastewater remediation as it is the most astringent tasting. Now let us move on to our methodology. In this research, we conducted experiments on 6 fruit skins and 2 types of tea leaves. To prepare the water extract, 50 grams of each fruit or tea was added to 100 ml of deionized water. The mixture was sonicated in an ultrasonic bath at 50 degrees Celsius for 1 hour to lyse the plant tissues and release tannins from the tissues. The resultant mixture was centrifuged and the supernatant was decanted for use. Next, we mixed 10 ml of each water extract with 10 ml of bovine serum albumin or milk solution before filtering the mixture to remove any precipitated protein. To investigate the feasibility of using solid fruit wastes directly for wastewater remediation, tests were also carried out using solid fruit or tea sample. The filtrates were used for 2 tests, the protein concentration test and the turbidity test. In the protein concentration test, the protein concentrations of the original BSA or milk solutions and filtrates after precipitation were measured using the Bradford protein assay to determine the relative amount of protein successfully precipitated and removed by each astringent fruit or tea. In the turbidity test, the turbidity of the untreated milk solution and filtrates after precipitation were measured using a turbidity meter. The decrease in turbidity of the milk solution after adding each fruit or tea water extract was used to determine the ability of each astringent fruit or tea in reducing the turbidity of milk. Now let us move on to the results. For the protein concentration test, the graph here shows the effectiveness of the different water extracts in removing proteins from BSA and milk. The effectiveness of each extract in removing either BSA or milk proteins follows a constant trend. Grape skin extract followed by mangosteen shell, English tea and mangosteen rind extracts remove the highest amount of protein. Solid fruit or tea samples were also tested to simulate the use of food wastes directly. The results follow a similar trend as when water extract is used. For the turbidity test, grape skin, starfruit skin and English tea were most effective, followed by the mangosteen shell. Mangosteen rind increased the turbidity of milk due to the mangosteen rind extract itself having high turbidity. Now let us move on to conclude the results. For the protein concentration test, grape skin, mangosteen shell and rind, and English tea showed the most potential to be used in treating proteinaceous wastewater. Although mangosteen rind is the most astringent tasting, it did not precipitate the most protein, thus disproving our hypothesis. This suggests that tannins are not solely responsible for the protein precipitating properties, and other organic compounds present in fruits and teas could also precipitate proteins. In the turbidity test, grape skin, starfruit, English tea and mangosteen shell were the most effective. Mangosteen rind increased the turbidity of the treated milk as its extract is highly turbid and hence may not be useful in proteinaceous wastewater remediation. To conclude, mangosteen shell being inedible and abundant as fruit waste holds great potential in proteinaceous wastewater remediation. Grape skin and English tea are not present in large quantities as waste and may not be as useful. The effectiveness of mangosteen rind in reducing turbidity could be improved by using its solid sample directly instead of using a turbid water extract. Now let us move on to the practical applications of our study. We have revealed the novel possibility of using astringent food wastes in proteinaceous wastewater remediation, where the use of waste to treat waste would allow for cost benefits and eco-efficiency. The proteinaceous wastewater could be first treated with astringent food wastes before being passed into conventional treatment systems. As part of our future work, different proteins could also be experimented upon as astringent fruit wastes might exhibit specificity in precipitating proteins. The excess of proteins precipitated could also be collected and reused as input material for fertilizers or animal feed. We hope that our study will spark new research possibilities in repurposing astringent fruit wastes to treat proteinaceous wastewater. thus achieving eco-efficiency in wastewater remediation in the industry. Thank you.